Hello all, it's the end of Monday, our first day back of this new semester. And if you're like me, you might be feeling a little tired, but I wanted to just do a quick, maybe 6-minute, we'll see, mini lecture on photosynthesis to help you guys out with your work that's coming up. So I will start by sharing my screen. And because of course, believe it or not, I've made some slides to try to help clarify things. And so I want to talk to you just really briefly about the evolution of photosynthesis. So plastids, what are we talking about here? Probably you're all familiar with chloroplasts, which are the photosynthetic beasts that we will spend the most time talking about. And those of you guys who were able to come to class on the last Thursday, we talked a little bit about some of these issues as well then, but there's a lot more we can learn because plastid is a generic term and chloroplast is one example. There are many other kinds of plastids. For example, I believe it's the etioplast, which is a plastid that's turned dark, is what makes tomatoes red. We also have chromoplasts, which is the plastid synthesizing pigments involved in photosynthesis. And we have the apicoplasts, which we'll be talking about in the coming days, which are present in apicomplexa like malaria. And there are many, many other kinds of plastids out there. So plastid is generic, and chloroplast that you're familiar with is a type of that. There are general properties of chloroplasts. They are double membrane bound, and chloroplasts, the one that are best studied, absorb light on chlorophyll and produce ATP. So my guess is you saw this in high school and perhaps early in college. Here is a picture of some plant cells with some chloroplasts within them. And here's a picture of what these chloroplasts would look like. And they have these membranes called thylakoid membranes, double membrane bound outer inner membrane, and these thylakoid membranes. And inside these membranes, much like you saw in mitochondria, are these proteins, photosystem 1, photosystem 2, and all of these proteins are passing electrons to make ATP. And this is what you learned about for the evolution of mitochondria. And much like mitochondria, when it comes to where do these come from, shockingly, unlike the nucleus and unlike the cytoskeleton, we actually have a good hypothesis. Where did they come from? Well, we think they came from cyanobacteria. These things with these inner membranes are not, are not eukaryotes. Here is a Synechochicystis, which is a very abundant marine cyanobacterium, and inside they have these internalized thylakoid membranes, and these thylakoid membranes are where these cyanobacteria make energy. So the hypothesis for plastids is that they came from cyanobacteria. The big mystery is not where did they come from, but how many times were they acquired. So we can all agree that there was at least some number of primary acquisitions, and here you have a eukaryote with its nucleus and its mitochondria, and it's taking up a cyanobacterium. How many origins, how many primaries were there, that is debated. But, and you can see the Larkham article that we discussed in class and that I'm asking you to write about. The current popular view is that there was a single primary origin of plastids. Why? The living plastids that have traditional genomes that we have studied so far seem to have conserved operon, so that's this linking of protein coding genes. They have conserved transit motifs in some, but not all. Dinoflagellates, for example, have plastids in mini circles that make no sense and follow no rules. They also have conserved transport systems. So red algae and green algae have this tick-tock system, which are the things that help transport across those double membranes. And at least many of the data suggest that there is a single group of reds, greens, and glaucophytes called plantae or archaeplastida. So that suggests this group emerged from the acquisition of primary plastids. And this was one of the major clades presented on. And I will say that at least another aspect of this whole thing is that this is a very popular idea that primary plastids happen only once in eukaryotes. And we'll end with this. And of course, since you're taking this class, you'll see that there are conflicting data. We'll return to that. And oh wait, actually we're going to dive into that a little bit right now. Here is the table from Larkham that I asked you to talk about. And here are these different groups that are argued to be primary. And if we look just at these guys at their chlorophylls, we see a diversity of chlorophylls. And when we go out to the secondaries, we see a greater diversity of pigments in these plastids. Sorry, I said chlorophyll, I meant pigments. So in terms of the arguments for the multiple origins of plastids, one aspect is the complexity of pigments. So what do we know here? We know that reds and glaucophytes have chlorophyll A and something called phycobilins. Green algae and euglenids have chlorophyll A and B. So that's okay. Maybe there was a cyanobacterium that had all of this. But as we get more broadly, we see that yellows, dinos, browns, and goldens have chlorophyll A and C. Wait, where did chlorophyll C come from? If these are the primaries, they only make chlorophyll A and B and phycobilins. And then that's just considering the chlorophyll A, B, C, and phycobilins. But if we go back to our table, and I'm sorry to go backwards, we can see among these things, there's also alloxanthin, fucoxanthin, pyridinin, diadeoxyxanthin, and so we have to understand where did all of these pigments come from. So here's just part of the story, and the question is there a cyanobacterium that has even just these three, A, B, Cs, and phycobilins? The answer is there's nothing alive, and if you add in all those other pigments, there are no living cyanobacteria that have all those pigments. So you have to imagine the super duper uber cyanobacterium in the past to explain the complexity of pigments, and I've shown just a few of them here. One other point here, if you go back to the Knoll paper on eukaryotic fossils, we learned that at least twice in the evolution of eukaryotes, there's been a period of Earth called snowball Earth, when the entire globe was covered by a kilometer of ice. And another big mystery in the evolution of photosynthesis is how do the photosynthetic lineages survive during snowball Earth? So we end up with these different hypotheses about algae, and I put algae in quotes because I don't know what an alga is. One is this picture that we have that I've asked you to write about from the Archibald paper, which suggests primary acquisition of plastids. I'll blow that up in a minute. Secondary, tertiary, multiple tertiaries, lots of complexity. These dashed lines are uncertainties. So here it is, one hypothesis that there was a single primary acquisition at the base of glaucophytes, chlorophytes, green algae, and the red algae. And I want to tell you who these guys are. Red algae, this is Porphyra. These are the band geomorphs for which there's a very good fossil at about 1.2 billion years. And if you don't care about eukaryotic fossils or don't care that much about them, the other thing we know about red algae is this is where nori comes from for seaweed. Green algae are so diverse. I pointed that out in the mitosis lecture. Here's a multicellular green alga. We'll meet multicellularity later. This is Volvox. It's multicell differentiated, has male and female parts, and gives rise to multicell babies. And then there's this other group. These are the three primaries, red algae, green algae, and the glaucophytes. These guys are economically unimportant, species poor, but they have these peptidoglycans still around their plastids. So the glaucophytes are the only eukaryote that still has peptidoglycan. When we think about all these secondary origins, there are multiples. How many we don't know. How many is debated. Sometimes, so here is the primary, sometimes we think a heterotroph engulfed a eukaryote to give secondary. Sometimes we think another eukaryote engulfed one of these secondaries to give rise to tertiary. And sometimes we think you have serial, where you have heterotrophs and where you have the multiple engulfments of plasmids. Basically, what we think is that it's very complex. Don't worry about the specifics here. But one example are the cryptophytes, which was also a major clade presentation. And in the cryptophytes, you have this crazy little flagellate here with two flagella. It has its own nucleus, and inside it is something that is clearly a red alga with a red algal plastid. And the reason why we know it's a red alga is it has a red algal nucleomorph. So go look at cryptophytes in our major clades. It has the red algal nucleus and its own nucleus, very strong evidence of secondary endosymbiosis. So these are the secondaries here that we're talking about, the cryptophytes that have a nucleomorph in them and all these other crazies here. And you, if you want to, you can see about the placement of these, but I'm going to let that go for now. So the one hypothesis was the chromalveolate. You had a primary endosymbiosis giving rise to reds, greens, and glaucos. Sorry, reds, greens, and glaucos. And these greens, this part is pretty clear. Greens are only found in euglenids and the chlorarachniophytes, green spider algae. These are rhizaria, these are excavata. The popular hypothesis at the time, and still held on by many, is that there was a single engulfment by a eukaryote of a red alga that gave rise to the chromalveolates, which included the alveolates, which we'll be talking about in the coming days. Here are the sterninopiles, and then these two lineages, the cryptomonad with the nucleomorph and haptophytes, which we won't talk about at this moment. And as I think you may have gotten from the major clade presentations, I don't believe in this hypothesis, nor do I believe the evidence supports it. So instead, we have this complex mess that I ask you to write about in your question for the coming week, where you can see this idea of maybe there was a tertiary here, a tertiary, secondary, serial, blah, 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 lots and lots of complexity here. And this, as I pointed out in class, Archibald dances between chromalveolates being true and SAR, which we're going to be talking about coming days, sterninopiles, alveolates, rhizaria. Here are these cryptomonads and the haptophytes out on the outside. And there's other trees. This one maybe we've seen in class before. This is from my work in my group, and photosynthetic lineages exist here in the Archaeplastida. We think haptophytes and cryptophytes are essentially orphans. We don't really know where they come. And within SAR, you have photosynthesis in here, and apicoplasts, we'll learn about them here, and photosynthesis in many of these guys as well. So I would end by saying that the secondary symbiosis is still debated. Now to close, I wanted to ask, is there evidence for multiple primaries today? I argued earlier based on Larkham that there's so many pigments, a single primary may not make sense, but how about extant examples? And there are two, and I asked you to mention in your essay either Rhopalodia or Pollinella, and to think about what they tell us about photosynthesis. So here's the answer in brief, but go look at Larkham for the details. Here's a diatom. Diatoms are photosynthetic. This diatom gave up its normal photosynthesis and acquired its own plastid independently from cyanobacterium. So this is the cyanobacterium that now lives inside this diatom. Clearly another independent acquisition of a cyanobacterium. And the other one that we talked about in class is Pollinella. Here it is. It is a shell-building amoeba in the Rhizaria territory and has these plastids in it. And the hypothesis is that a shell-building amoeba engulfed cyanobacteria to give rise to this genus named Pollinella, which is photosynthetic. And so in the end, we have these two examples, the shelled amoeba and the diatom, that have independently acquired photosynthesis. What some people argue is, well, it's not really a true primary because there hasn't been the same level of integration of the genomes as we see in green algae and red algae, so it's not true primary. I leave those semantics to you. And so on the final, I may well ask a question, a very broad question about what is the evolutionary history of photosynthesis in eukaryotes. I also invite you to watch this video, which is on Moodle. I've pointed out which chapters I think you should watch, both in the slides and here. And it's this amazing man named Stuart Pickett Heaps, who loves diatoms more than anything. And he starts us off by flying over diatoms in a helicopter. So I have pointed out some of the chapters that I think would be worth looking at. And I think one of them includes, sorry, that's rude of me. One of them includes diatom sex and how they have sex in a glass box. So it's almost like a shoebox. Well, I'll end with that. So you can watch this. Here they are flying over a field of diatoms.
Hi, I'm Jeremy Pickett Heaps, and we are flying over the countryside near Lompoc in Southern California.
Okay, so I think I blew his name. It's Jeremy Pickett Heaps. And so I will end by saying, take a look at that video, because diatoms are like shoe boxes. So imagine a shoe box, and if you open the box up, the top of the shoe box makes another bottom, because when they divide, they make an inner, but the bottom of the shoe box makes an inner that is smaller than the original top. So over this course of a season, diatom populations on average get smaller and smaller, because then the next generation opens up and everybody makes an inner. So they keep getting smaller and smaller, the average population size. And then they have to reset by having sex when the cells burst forth from their little glass boxes and reset their size. It's really cute. It's very relaxing to watch this video, and I urge you to do so. Take care all.